The main goal of our research is having a new clinical application, which will be human spermatogonial stem cell autotransplantation. And this technique will help many patients, especially the cancer survivor boys, the immature boys who already banked some testicular tissue before starting chemo or radiotherapy. And by doing this transplantation, we will be able to help them to restore spermatogenesis by using their own materials. To having this new clinical application, we need to address several concerns, and actually we are very happy that right now, thanks to the result of our research and other groups, we are able to harvest testicular tissue very safely from their testes. We can freeze them down. We can isolate the spermatogonial stem cells from frozen tissue. We can in vitro propagate the stem cells to have enough number of cells for transplantation in the future. But still, there are some steps that we need to go through them, and one of the most important thing is the safety of using these cells for transplantation. And the safety is very important because we are using material from a cancer patient. If there is any contamination of malignant cells in the testicular tissue, then there will be a chance of reintroducing cancer to the patient. That will be terrible if it happens. Then we have to be sure to remove all the cancer cells, or to be sure that there is no cancer cells contaminated the testicular tissue from this patient. This is the main aim of the current study that we just published in Fertility and Sterility. And in this study, actually, we decided to choose acute leukemia as a model because it's very common in children. These children, they have a very good survival time. And on the other hand, there is a high chance of infiltration of leukemia cells to the testes. Then it should be a good model. What we did, we tried to culture the leukemia cells from 3 different patients in the same culture system that recently we established for in vitro propagation of human spermatogonial stem cells. And interestingly, we found that these leukemia cells, when we culture them alone, they are dying in 2 weeks of culture. After 14 days, we didn't see any alive leukemia cells. Next step, we decided to mix the leukemia cells with human testicular cells in different concentration and put them in the culture and follow them, monitor them, what will happen. We found that even when we contaminated the testicular cells with 40% leukemia cells together with 60% of testicular cells, after 26 days we didn't find any leukemia cells. And the way that we tested was using a very sensitive clinical PCR, which we called MRD PCR, minimal residual disease PCR. And with this PCR method, we could detect 1 out of 10,000 or 1 out of 100,000 of cells if there is any malignant cells, which is very sensitive. And as I said, there was no malignant cells after 26 days. And we already showed that we need to culture testicular cells to propagate enough spermatogonial stem cells at least 35 days. And this means we are fine, and if we culture them, we will propagate the spermatogonial stem cells and we can remove the leukemia cells. But we call this a study a pilot study because we just tested in 3 different patients, and also there could be some other leukemia cell line which are more aggressive. Then it will be good to try more patients and other types of cell lines and see if we have the same result. Because this is very, very important issue for the safety of future human spermatogonial stem cells autotransplantation.